Development of neuronal circuitry of the human prefrontal cortex during the last 2 months of gestation. Structural organization. During the last 2 months of gestation, the frontal cortex and related subcortical nuclei grow rapidly. All primary gyri and sulci are recognizable at the cortical surface, figure 2.1f, as demonstrated by in vivo magnetic resonance imaging, which, in comparison to postmortem material, provides a more accurate picture of convolutional development. However, in classical neuroanatomical studies, it was not fully appreciated that, at this age, laminar and aerial cytoarchitectonic organization remains surprisingly immature. For example, the transient SP subplate is still interposed between growing white matter and six-layered cortical plate. The six-layered cortical plate was described by Grundtypus as Grundtypus, Grundtypus, by Brodmann in 1909. In this six-layered pattern, figure 2.1f, one can distinguish individual layers, but cell sizes, morphology, and aggregation within these layers are significantly different from those in the postnatal cortex. First, layer 3 pyramidal neurons are small, and characteristic sublayer 3C cannot be delineated. Second, layer 4 is densely granular, figure 2.1f, and this granularity continues into premotor and primary motor cortex of the frontal lobe. Therefore, the criterion of layer 4 granularity, for example, agranular, dysgranular, granular, cannot be used to delineate frontal cortical areas as in the adult brain. Third, layer 6 is wide and very cellular, with gradual transition toward the SP subplate. Fourth, layer 1 contains numerous large neurons, many of which show the characteristics of Cajal-Retzius cells on Golgi preparations. That's Cajal and then Retzius. The most obvious sign of immaturity is the presence of the subplate, SP, which can be demonstrated with some simple staining techniques such as PAS Alcian blue staining. Capital P, capital A, capital S, dash, capital A, and then LCIAN blue staining. However, the SP subplate gradually diminishes in size after 32 to 35 postconceptional weeks. The substrate for the gradual diminishment of the subplate on magnetic resonance images is the decrease in the amount of extracellular matrix and other growth-related guidance molecules. The decrease in thickness and signal intensity in the subplate SP is the hallmark of cerebral cortex on magnetic resonance images in the preterm infant. The ingrowth of long cortical pathways into the sublayers of the CP cortical plate is another important factor in diminishment of the SP. Parallel to the decrease in the size of the subplate, there is development of corona radiata composed of thalamocortical and other projection fibers. The corona radiata fibers form segment 3 of von Monachov, 1905, and continue in gyral white matter. But tight connection of the gyral white matter with cortical layers is interrupted by existence of the subplate. Neurogenetic events. Data on neuronal proliferation in non-human primates obtained in stages comparable to human development indicate that we cannot expect production of new neurons in the prefrontal cortex of the late preterm infant. The notable size of proliferative ventricular, subventricular zones and ganglionic eminence are most likely related to proliferation of astroglia and oligodendroglia. The presence of areas of immature cells around the anterior horn of lateral ventricles, which are characteristic for frontal lobe, probably reflect waves of cells on the way to more superficial cortical layers. It is also very likely that some late arriving neurons still migrate because the migratory radial route is longer in the prefrontal cortex than in other cortical regions. Growth of dendrites is a significant event in preterm infant. This phase of differentiation is influenced by glutamatergic thalamic afferents and is described as a second phase of dendritic differentiation in the prefrontal cortex. Parallel to this event, there is initial development of characteristic dendritic orientation in non-pyramidal local circuitry neurons. The most robust neurogenetic event in the preterm infant is growth of long cortical pathways. Callosal fibers are in the phase of growth around ventricles and show significant increase in number, described as exuberance. There is no reliable chemical marker for direct demonstration of long cortical associative fibers. The DT tractography demonstrated preliminary results without identifying sources of associative pathways described in adult prefrontal cortex. So assuming DT is diffusion tensor, the reference title is Efferent Association Pathways from the Rostral Prefrontal Cortex in the Macaque Monkey. Petrides and Pandya, 2007. DT tractography, diffusion tensor imaging. The conventional MRI shows periventricular zone rich in extracellular matrix, which corresponds to axonal strata containing long associative fascicles such as fronto-occipital and superior longitudinal medial fasciculus, described by von Monikoff, 1905, in developing fetal and child brain. And then that author's name is VON space MONAKOW. In addition, fiber bundles that contain efferent associative pathways from the rostral prefrontal cortex, such as external capsule and cingulate fasciculus, are well developed in the preterm infant. Functional organization. There is further maturation of evoked and event-related potentials. The gradual appearance of synchronous EEG activity and slow activity transients become less frequent. Analysis of general movements that depend on transient circuitry shows characteristic quote-unquote writhing general movements. Because of the significant synaptogenesis in superficial layers of the cortical plate, cortical dipole switches from predominantly deep to predominantly superficial generation of electrical activity. Reference Molliver, 1967. Accordingly, preterm infants show surface-negative cortical potentials that change to surface-positive potentials during the early postnatal period. As stated for early preterm infants, the significance of sensory input for maturation of the prefrontal cortex is not known. In experiments performed in monkey fetuses delivered 3 months before the term and exposed to normal light, intense visual stimulation did not affect the rate of synaptic production. The reference for that is 1989. Accordingly, one can assume that fetal afferent input to prefrontal cortex before the birth will not affect the rate of synaptic production.